A few more seconds because there's usually a buffer. 1 minute. Yeah, that makes sense. Are there people on? Okay, I guess we can probably get started. Good morning, everyone. We're very happy to have Hasindu today. Before I guess he gives his talk, I want to introduce him. So Hasindu is currently a lecturer at UNSW Sydney. But essentially for those who are not really familiar with these titles, I guess this is really an equivalent of assistant professorship as in the United States. He received his PhD again from the UNSW Sydney in 2020, and in between his current position and the PhD, he was also a senior scientist at the Garvan Institute, where he's still a visiting scientist, I assume there. And you may know him mainly from his works on nanopore signal analysis and also A5C and many other works that he did on this area, as well as basically designing hardware and accelerating genome analysis, which is also something that we're quite interested in. But I know Hasindu basically from many different occasions. I remember seeing mainly Hang Lee's tweet and also basically your work on SLO 5, where I guess he was one of the people who challenged the community to think about like what sort of different file formats we should be using especially for nanopore signal analysis, which is basically quite needed in the field, and also he has done a lot of other great works on that domain. And of course, like based on these works, he received several awards, which I want to basically read them as I don't remember from top of my head. He is a recipient of the ARC Discovery Early Career Research Award in 2023, and also a ABA CBS, I guess, Outstanding Bioinformatics Software Developer Award recently in 2024. So we're quite happy to have you, Hasindu, here. And with that, please take it away.
Thank you very much, Ken. Okay, so I'm going to talk about scalable, efficient, and real-time analysis of long-read genomic data. And what's there is a portable MinION nanopore sequencer with a flow cell and some sample also loaded. It's very portable. I could have brought it, but in case run into troubles in the airport, didn't bring it, just put a photo here. Okay, so a bit about myself. I did my bachelor's degree in computer engineering in Sri Lanka. In the middle of Sri Lanka, there's a town called Kandy, and there we have University of Peradeniya, a tropical green university, quite beautiful. And then for my PhD, I moved to UNSW Sydney, and there I happened to do a project with an intersected area of genomics and computer science. That's how I ended up in bioinformatics. So now I'm working as a lecturer in the School of Computer Science and Engineering at UNSW Sydney. And in parallel, I am a visiting scientist at Garvan Institute of Medical Research, which is also in Sydney. Okay, so this is going to be the outline for my talk today. First, I'm going to give a very brief background on sequencing and analysis. Then I'm going to talk about few example computational methods we developed to enable scalable and efficient and also real-time data analysis. And finally, I will quickly go through some ongoing work too. So before going into this section, can I know if you are familiar with genome sequencing? If everyone is familiar, I can go very fast. If everyone is if someone is not, I can give a brief introduction. What do you think? Yeah.
There are still people who are questioning what
okay, so yeah, okay, so I'll spend maybe 10 to 15 minutes then to go through the background quickly. Okay, so DNA and RNA is like the blueprint of life, we say. Thinking about a computer program, just like that. DNA will have all the data and the instructions to create and make a living being function from the scratch, self-executable. So then usually this DNA has genes and stuff which gets transcribed to what we call RNA, and then those get translated to protein, which creates a living being. So because DNA is the blueprint of life, obviously reading it will have a lot of impact on a large number of fields. One of them is in medical, and every other fields involving living organisms, like forensics, ecology, basic biosciences, many. And therefore, it's very useful tool to go and read the genome. So you may know that the genome is made up of 4 types of bases, especially DNA, like ACGT bases. Okay, so in a human genome, the DNA strands are pretty long, and the whole genome is 3 billion bases, 3 gigabytes if you put in ASCII. Well, compressed might be 700 megabytes. In addition to these bases, you may have heard there is these things called base modifications. C can be sometimes methylated. And that's not the only type of modification. There are loads of other modifications in both DNA and RNA. So that we collectively call epigenetic modifications. And those are also known to be pretty useful in a lot of applications. Example, in medicine. For example, we know that methylation patterns can have applications in detecting tumors or classifying tissues, detecting biological age. So those can be very useful in a multitude of scenarios. So genetics, that's the DNA, which is hereditary, mostly coming from your ancestors. Whereas these epigenetic modifications are not hard-coded. Depending on the environment and time, they can modify. So that together with the genome creates an actual living being. So now let's go about reading the DNA. In 1990s, the first types of automated sequencers came, semi-automated, like the Sanger sequencer, was very large and lot of manual labor and very costly. And with those technologies in 1990s, the first human genome project started. So it took a decade for a number of scientists to sequence this first human genome. Lots of money, obviously, millions, billions of dollars. But then in 2000s, here came an automated sequencer. So with that, you could do the sequencing of a human genome at a much lesser cost and faster response time. Within a few days, several $1,000s, you could do it. But these were giving very short rates. I told you the genome is like 3 billion bases, and a chromosome can be like few megabases, like hundreds of megabases. But these reads are very short, like 100 to 100 bases. So as you can imagine, it's very hard to resolve the whole genome by using these like Illumina, like second generation sequencing alone. But recently, these third generation sequences have come, and they provide us with long reads, like 10,000, 20,000 bases, or even more. Now, the good thing is you have a bigger picture of the genome. Of course, not perfect. Would have been ideal if you could read the whole chromosome, but so far we don't have such technology. But at least this is better than nothing. With these kind of long read sequences only, like very recently, like I think 2 years, 3 years ago, like 8% of the gaps in the previous genome project were completed. Even though that 1990 human genome project finished in a decade, they could only do 92%. There were like complex repetitive regions that were not resolved. So only recently, like several years ago, they fixed it. So you can see that's a very big advancement in that term. So today the talk is mostly going to be about nanopore sequencing because it's like third generation. It can sequence long reads, and the other benefit is with the sequencing data, you can also get those epigenetic modifications I mentioned. So 2 in 1. Good. And also there are portable sequences, which is very nice. So currently Oxford Nanopore Technologies is the company that makes nanopore sequences. But of course, recently BGI, MGI from China also unveiled a nanopore device. We don't know too much about it at the moment, but competition is always good in my opinion. But at the moment, seems like nanopore, like Oxford Nanopore is dominating the area. So they have multiple line of devices. So on this side, we have portable devices, like Minayan, you can put it in a pocket. And then we have the P2 Solo, which is like a brick. You can still carry it in your bag. And then you have the Gridiron, which is like a bit large benchtop device. And then the Promethean, a larger benchtop device. So this one is quite good for doing bacterial and small genomes, whereas this one is good for even a human genome. You can do 2 human genomes in parallel sequence. And then on the other hand, this one is a monster. It can do 48 human genomes in parallel. Okay, so now let's go into how the nanopore sequencing works. So let's say that's the Minayan. Inside it, we have this consumable flow cell, which we load the DNA sample. And then on it, there is a panel containing hundreds or thousands of tiny little pores called nanopores. And it's like this, and see, this is a video that shows how a DNA strand passes through the pore. And when that happens, it gives the current signal. So if you look at here, there's a pico range ammeter here. So based on the characteristics of the bases, A, C, G, or T, the current level can vary. It's a very noisy signal, but today with the advancements of deep neural networks, we can go and get accuracies close to 98, 99% in average, which was 70%, I think 8 years ago, when they used hidden Markov model. So a huge improvement recently with neural networks, plus improvements to the actual chemistry and the pore to reduce the noise. Okay, so the other thing is, there will be slight variations in the current if the bases are modified. Remember I told you about epigenetic modifications? If it is modified, there can be slight variations in the current. So this is helpful for us to, detect the modifications. So raw data, the signal data is what's usually output from a sequencer, and they are pretty large. For a single human genome, it's like 1 to 2 terabyte at 30x coverage. Every position covered by 30 reads. And this data can be used to base call or modification call using algorithms. So this is what a single raw signal, a part of a raw signal looks like. So this is the DNA sequence, and this blue line is the noisy signal. Okay. And here I have individually put the k-mers. K-mers is like a small moving window. This nanopore, when there is a thing inside, it's not narrow enough to contain one base. So typically a k-mer, 6 bases, for example, it can be different based on the chemistry, affect the instantaneous current. So as you can see, if you have a moving window here, and then each level here corresponding to that particular k-mer. So in classical analysis, we have this thing called event detection, whereas we look at the signal for sudden jumps and then assume that something changed. But here you can see it's over segmented, the red color line here, see? But then we can use these events and align them using dynamic programming so that we get this true annotation here. So you could see that there is a good difference here, but here it's very difficult to resolve because it's very noisy and the amount of current change per k-mer is low. And this is what we called a POE model. It's like a table telling what each k-mer should actually give. So Oxford Nanopore has provided us with some tables which can be used for various classical analysis. So that's a very brief introduction into what's out there. So before we go into the next part, can I ask you if you have any questions? Okay. Okay, so now let's talk about the actual research. So what I'm doing is containing twin goals. One of the goals is to make sequence analysis portable. We have the MinION which we can carry on the pocket. But if we have to rely on a desktop computer, that's not great, right? Because you can't carry the desktop in a pocket or even a bag. Of course, if you are very fit, Jim, you can carry it probably 20 kilos or something. Yeah, but at least I can't do that. So I consider it not to be portable. So it's ideally good to have something like a laptop being able to process that data. Of course, in a real-time scenario, the data coming from the sequencer, if we can process while it is giving out, ideal. So that's one of the goals of my research. On the other hand, we have this Promethean, which can sequence 48 samples on a benchtop. But to process the data coming from it, you need a cluster of computers. You have those clusters, right? It's like a large supercomputer containing number of nodes. So if you have 4 Tesla V100s on each node, if you have multiple dozen of nodes, probably we can keep up. For a benchtop device, you need a whole institute's cluster, which is not again great. So ideally, we want to make the compute optimized so that we can do it on a reasonable sized, like a workstation. That's another goal. And of course, these two are related. If you optimize the software to be like, run on portable, then will eventually help like making that goal as well. So they are a bit coupled. And all of these, we are committed to provide open source implementations, which is the norm in bioinformatics, and good for science, like transparency, rather than hiding things in a black box. Okay, so like, I think in 2023, when I presented virtually, like with GAN, so I presented about the SLO5 format that we developed, which was a fundamental game changer that made like a bottleneck resolved. And back then, ONT used a format called FAST5, which was like very like, slow and a number of problems. So we demonstrated that you can get like 10 times better performance with SLO5. And then, of course, later then they like developed something called POD5, which is the like file format now. Which is also having some like, performance and other issues which I presented last time. But anyway, that's not the like, target today. Also to add, we have a wide range of ecosystem around SLO5 now, like libraries, tools. For example, here you could see that multiple languages are supported, and there are a lot of tools that support SLO5 format, including from like Safari Group. So today I'm going to talk about like some additions to this ecosystem plus other sequence analysis optimizations we did. And the first one I'm going to present is how we enabled population level analysis of signal data. Okay, let's see what that means. So, like today there are these like 1,000 genome data sets for nanopore, like publicly available data sets. And many people are generating such valuable resources too. And each human sample, if it is like 1 terabyte, for 1,000 genomes, how much is it? 1 petabyte of storage. So these are typically stored on like, accessible servers like AWS buckets or other servers. Now assume, right, base callers always keep updating and new modifications are added to the base callers. So quite often we would need to rebase call in the future. So let's say in a given study, we want to grab a certain number of genes which we believe to be important. And we want to base call or modified call these and do for downstream analysis. So traditionally, what do you have to do? Like you have to download each data set one by one to your like local supercomputer cluster or something, and then base call. And like downloading 1 terabyte, you may have tried, it's not that fast. So this process will take weeks. So to solve this, issue, we introduced the SLO5 curl, which is based on a simple idea. On a remote file, we try to partially access the reads that are only relevant to this genomic region. And that way, if you need like few hundred genes, instead of downloading 1 terabyte from each sample, you have to download like 1 gigabyte, a reasonable amount. So that way, you can quickly download within a few hours and also get the base calling done on those subsets. And importantly, it will save the like amount of money you have to pay to AWS too, because they charge for like egress or whatever, right? So, yeah. So let me now tell you how SLO5 curl was implemented. It's a very simple idea. So here, like this green color ones are the server side. This red one is the client side, and these yellow ones are like processes. I'll go through them like in a flow chart. So in the remote server, let's say we have a like a large SLO5, is the binary format of SLO5, if you don't know. located. And then let's say we have an index that gives like every read. Okay, here is the read, like that, it's an index. And then, like, let's say the SLO5 client on your computer asks for a read. So the first step is it automatically downloads the index from the server and cache it the first time, because next time you can reuse it. And then for a given read, you send the GET request to the server with the byte range, then you get it. And likewise, in a loop, you can get all the reads you want and then decode it. Okay, so the index loading happens only once. So it's in memory until the program finishes and get all those reads. So this is how we implemented it. And results are pretty great, even though the implementation is simple. So we tested with like 90 human data samples from the pan-genomic project. At that time, the 100,000 genome project wasn't there. So these genomes are like not sequenced at 30x, it's like 10x. That's why like 90 samples are not 90 GB instead of 90 TB, it's 20 TB. So what we did was, like, onto the local computer, we grabbed signal reads belonging to like 100 genes from all these 90 samples. We used a combination of SAM tools and SLO5 curl for this, because like most of the time you save the old base call data there too. And SAM tools, if you compile with the correct options, you can get like genomic region. So we do that, get the read IDs, and then feed it to SLO5 curl, and then finally we base call everything. So everything fetching from the internet and base calling with super accuracy took like 6 hours. Well, the internet connection was like, I think 1 gigabyte per second. We used a university connection. But still, if you did that in the classical way, good luck. It will take probably a year. And not to tell, it breaks the connection every now and then. Okay, so like, wrong side. Okay, so this is the amount of time taken for each sample. The x-axis is the sample, y-axis is the amount of time taken for like SAM tools and SLO5 curl. You could see that per sample, getting 100 genes takes only like few minutes. Yeah. So if you want more information, it's available in Giga Science, and the work was led by Bonson. And the next one I'm going to talk about is a new compression strategy we introduced for nanopore signal data. Like, we wanted to try to minimize this 1 TB size a bit, because it's a lot of storage. So we introduced something called the XCD. If you are curious what that means, it stands for exceptions zigzag delta. So with that, we tried a number of available, like compression algorithms too. And like we have plotted it here. And then you could see that XCD like gives the current best compression ratio. The x-axis is the compression ratio. So XCD, on top of the encoding, you can apply either ZSTD compression or zlib, like we had for SVB and other stuff. So compared to Oxford Nanopore's VBZ, we have like around 2 to 3% improvement, not much. But the reason is like we have seemed to like kind of reach that like saturated point where like noise in the signal cannot be like compressed, because it's white noise. Usually white noise cannot be compressed. So we believe that we have come to that saturation point. So with that, we wanted to explore lossy compression, because if we get rid of the noise somehow, we could go much better. So for this, what we used was this fact. So this is a histogram of signal samples coming from a Promethean dataset. The x-axis is the raw signal values. Like analog to digital converter gives digital values, and those are the values. And y-axis is the frequency, like how many times that particular value occurred. Ideally, we would expect like a bit smoother, one, right? If the ADC was nice. But here we see that like sometimes there are spikes, dips. It's a bit strange. Like if you zoom into this, like we observe that most of the time this up and down thing happens when things transition from 1, 1 to 0, 0. So likely to be something with the ADC. Yeah, we tried to get some information from nanopore about the ADC they are using, but apparently that's proprietary things. But anyway, that's likely to be like some sort of noise. So if we can get rid of this noise, then we could achieve better compression. Of course, there's a lot of sophisticated like noise removal techniques, but first we wanted to see the worst possible noise reduction method and see whether it impacts the accuracy. Okay, so what we did was we tried to get rid of like bits, the least significant. So this is the original histogram, okay? So we remove the least significant bit. How we do it is rounding to the nearest 2. And you could see that noise slightly reduced. And then we remove 2 bits. So in that case, like 2 to the power 2 is 4. So we round off the signal values to the nearest 4. And then you draw the histogram. So you could see around 8, when 3 bits are like eliminated, we kind of get a smooth like histogram. Then we like checked how it impacts the accuracy. So you could see that here, this is the number of bits retained. 11 bit is what we originally have in a data set. Then we removed 1 bit, 2 bit, like so on. And we included super accuracy based calling and high accuracy based calling, both Dorado and Guppy, 2 fish based callers from ONT. They always name tools with the names of the fish if you don't know. Dorado is a huge fish. Guppy is a small fish. Okay, so this y-axis is the identity score. Like after you align the reads, we calculate this metric. It's like how much it differs to the expected. So you could see that like initially, there's no much change to the average like mean read accuracy. And after that, it crashes, which means that here we seem to be like losing the actual information. So then we did another test for 3 bits removed data. Like 11 bit, this is the original. This is the 8 bit. Both base callings were run on the same GPU. And per each read, we plotted the, the correlation plot. And here you could see that, like, of course, like they are not exactly the same, but slightly different, but they are mostly along the diagonal line. If they were identical, you should get a single line. But on the other hand, this one is with original data based called on a A100 versus V100, 2 different GPU architectures. You still don't get the same base calls anyway, probably due to floating point and all the other, like, non-deterministic nature of floating point calculations in a computer. So you see, like the spread looks very similar. So we believe that if base calling on 2 different GPUs can give that much spread, probably like this is like fine too. Okay. And yeah, we did a number of other tests, including like seeing if the methylation modification accuracies are affected, whether RNA is affected, did a lot, but we couldn't see much like degradation to the accuracy. The results are pretty good because you could see here that like there's like 44% serving with the state of the art, 0.5 if you remove like 3 bits, like 8 bit. And the performance like was also like not different, almost the same or even better. Like here, like XCD is with ZSTD, was what I'm talking about. And then this is the VBZ. Like I did a test where I decompressed and compressed the data set. So you could see that times are even a bit lesser with similar amount of RAM. So with a very like naive bit reduction method, you can still get like good accuracy. So we believe that with sophisticated noise removal techniques, you can do even better. So yeah, these results are like now available in the like genome research article, which is only available, I think, in a couple of days. Currently, it's like the, what is it called? I don't know, without the proofs or whatever. So the work was done by Hasindu. And the next one I'm going to talk quickly about is simulation and visualization of signal data, even on your laptop. This is mostly targeted towards developers of signal analysis tools. Having a simulator and a visualizer is a great way to start with than real data. And visualization is good to, you know, see what's happening, to see if we can find anything interesting. So for this, we like develop this Squigulator, which is a very easy to use tool, because I believe a simulator should be much easier to use than getting actual sequence. If it is harder than, you know, installing is harder than getting a sequencer and sequencing, no point of that simulator. There are many such like, you know, simulators like that, which we tried to install after 2 days, gave up and just wrote our own simulator. So this one is easy to use and also supports multiple chemistries. You can give a try if you are a developer, and more information is available there. But please be noted that it's not a replacement for real data, but this is like, you know, simulation data to start with your project or to debug, you know, you could have, like have a truth set. And for various things, you can use the Squigulator. Squigulator is the one we generate. This is the visualizer. So it has multiple options. So one option is visualizing like the alignment of a signal to a reference like read, like a base called read. So you could see that there are two modes are supported. One of them is time scale, like the actual signal we get. You could see that some of the bases are like stretched, some are squished. That is the reality. You can't control the speed of a thing going through. And this is like another like method we support where the user can ask the thing to be equal, like squish it and make it equal. The most interesting feature would be the pile-up view. Like we have the pile-up view IGV, we have the pile-up view in the signal domain now. So here, this one is everything drawn on top of each other, and these are individual reads. Okay, and here, just for the comparison, I have provided Squigulator generated ideal signal, because Squigulator, the simulator supports multiple options with noise, without noise, different levels of noise. For comparison, this is a step function with no noise. You could see that the real signals here have quite good resemblance to what we have here. So these kind of tools are pretty useful in like discovering certain new modifications. So here you see this one is an example where some of the reads are methylated and some are not. You could see that, right? Some like, you know, have a higher signal level than other. And sometimes the base caller does mistake thinking it's something else. So yeah, so for more information, you can refer to the publication and try the tool. And also the most recent work we also got was like improving, like creating a new pore model. Because like when a new pore model, like a pore comes, like R10, R9 or whatever, like this method can de novo generate a pore model. So yeah, so anyway, like for the interrupt of the time, I will quickly skim through some of these examples without going into too much details. So this example is where we implemented the real-time methylation frequency calculator for the Minion and Promethean. So the idea is like pretty simple. Rather than waiting at the end of the sequencing run, we in real time compute the methylation frequency using a software called RealField we implemented. So this is like the architecture. So it's implemented using a bunch of shell scripts and a C program. So when a data batch is ready, it will do the processing and keep that in memory. And when the next batch comes, it updates. So this allows a custom program, like a adaptive sampling, like a implementation to query and see whether we have got enough methylation or something, and then do the experiment accordingly. So if you are interested, you can so this is the real-time graph. We just showed that for a Minion, we can keep up on a laptop, and on Promethean, we could keep up on a desktop. So more information is available here. And finally, these are some recent hardware acceleration work we did. Some of them, most of them were targeting this chaining algorithm in Minimap. So this is away from signals now, only aligning bases. So chaining is one of the fundamental algorithms in sequence alignment. And I think the signal alignment method which can develop also has chaining in it. So a fundamental operation. So here we try to accelerate this process using FPGA implementation, and also using a RISC-V custom instruction set. So the idea is we would be able to in the future get a chip or something with all this work, but it's a long time from now. Yeah. So, and now I will quickly show you some ongoing work if you are interested. So one of the software we are currently implementing is Dorado, which is a lean base caller to Dorado. Why do we have it? There are quite a few things I find a bit difficult with Dorado. One is it's having too many dependencies. So compiling is a bit hard. And the main reason is, even though Dorado is open source, there is a part called COI in it, which simply downloads a binary from the nanopore repository and link it. Okay. So when you want to install or run Dorado on a device, GPU that's not officially supported, let's say an old GPU. If you have A100, B100, H100, 3090, all fine. Yeah. But I wanted to make sure people with older GPUs also can, do some base calling, because not all the countries in the world have that much resources to afford the newest GPUs. For example, in Sri Lanka, I know that we still rely on some older generations of GPU. So wanted to give support for multitude of things. So that's why we were implementing this open source version of whatever this COI does, which we call OpenFish. And with this, we could support tiny Jetson boards. And also recently we enabled support for the AMD GPUs. So I think ETH has a AMD cluster, right? The hack cluster. So you guys can try if it works. Still under active development, but the base calling with V5 models can be done. Of course, don't expect it to be faster than Dorado. Still long way to catch up. Still, it's not too bad, 2 times to 3 times slower. We could show that in the Pausi Supercomputing Center, which is one of the largest supercomputing centers in Australia, which harbors these AMD GPUs, like a human genome 20x, we base called within 24 hours using 4 GPUs. Yeah, so I think it's still reasonable. Whereas 4 A100s using Dorado would take I think 8 to 10 hours. Okay, another work we have been doing is this Conator project. We are trying to assemble genomes in real time using adaptive sampling. We are working towards that. A way to improve the accessibility to telomere assembly. Rather than using tens of flow cells, we try to achieve it using a couple of flow cells. And finally, we are developing like a mini mod, like a simple tool for like calculating the methylation and other modification frequencies. So this was inspired by that real freak I mentioned. The implementation we did turned out to be very efficient. So we decided to make offline tools too, so that people can take advantage of that if they want to run offline. So anyway, like that's all I had to present today. I should give a big thank you to all of the people involved, including the students, mentors, collaborators, not just from UNSW and Garvan Institute, but across multiple institutes, which the list is too big to put here. I have put the names of some key personnel, but the actual list is very long. So thanks to all of them, we have this stuff that I presented today. So it's now time to get some questions.
Right, thank you. Very nice presentation. Many nice ideas. So I have a few questions. One is one motivation for your curl approach was essentially you might want to recall data, right? So
Sorry?
I mean, you had this curl setup where I can essentially just index and then I mean. Two questions on this. One, this works well for human genomes where you have a coordinate system, right? So and then the premise is that you can recall data, right? So you think this is something advisable to do in a sense? I mean, let's say ONT changes chemistries all the time. So are all the newer callers backwards compatible? So, or is this essentially something that at some point we might not be able to use the old data anymore?
You mean slow five?
No, in general. I mean, just because I mean the translation from raw signal into base space, right? That makes assumptions. You need to train a base caller.
Yeah.
And ideally, on newer chemistries, you retrain the base caller, right? So and that means if you want to integrate many different data sets that have been produced with many different chemistries, you might run into some issues, right? So.
Yeah, I mean, like, but so far, like it's possible to base call like most of them with like the existing like tools, so as long as like ONT provides access to these older versions of Guppy and Dorado, like we are able to base call them.
Okay. But the second question on this is, so now you need a coordinate system to index, right? To build your index on the BLO5. Could you also imagine to index the raw data? For instance, let's say this is not a human genome, this is a metagenome, right? And I essentially would like to find signals that are like the one that I already have, right? So this goes a little bit in chance direction, probably with the raw indexing. So
So I think like this work at the moment we presented is searching like just based on the read ID, but I think what you are like targeting is something like a searching the actual signal. I think like, I mean, definitely looking at how these sequence spaces work and how things have translated to like signal space, definitely possible. But of course that would involve like a lot of algorithmic advancements. So like, I think showed that like you can do even assembly in the signal level, right? Raw sample. So yeah.
Okay, last question. You showed this squiggleizer. Can I plug this into the curl? So essentially, I just want to look at a few regions that are in part 5 files online. Can I do this? In a sense, just can I fetch directly into the visualizer and look at the files?
Okay, so yeah, like there are some steps involved because you have to align the like data to a reference to. So I have given like we have given the the tools that you have to run one by one with examples. So if you follow those instructions.
Okay. I had somewhat assumed you already have the alignment, because otherwise you wouldn't have the reference coordinates for indexing, right?
So is this about the squigglizer or the?
I mean, both, right? So in a sense, for your curl, you index on a reference-based coordinate system. Hence, if I get signal, I already know where they would belong. Hence, I also have the notion of whether they align to a reference. Hence, you can use this to directly
if you have that like alignment data, you can.
So I was envisioning something. I just have a web page, and I essentially click on a few resources, and then I essentially it pulls. It's like a genome browser, just for files.
Yeah. Right? So I mean, at the moment, of course, squigglizer is like not like implemented to do it in a browser-based setting, but like the implementation is definitely something like possible.
But logically, this would work, right? So you could...
logic works as that was the plan actually, right. But you know, like a lot of implementation work.
I know how this is. Okay, cool. Thanks.
Thank you very much. A super impressive talk, Hasindu, and it's amazing to have you here in person, such a contributor to the nanopore technology. I would actually have a few questions about the Cornetto tool as well that you just briefly presented, so maybe I quickly come down afterwards. So about maybe the quick questions about the talk. So this Lorado tool, like for super amazing idea, by the way, to make Dorado full open access. Do I understand correctly that you wouldn't have to retrain the base caller? So you're basically using the trained Dorado, the model, and you're implementing it differently.
Fortunately, like Dorado like uses this PyTorch lib, which is the C version, C++ bindings to those models. So it's in a format that's already open. So thanks to that, only we could do this. So we don't need to retrain, like we can just get the Dorado models and use it. So we have like tested and supported like most frequently used base calling models.
Fantastic, thanks. And maybe one more question also about the slow 5 curl that Andre already, I mean, I have a few questions, but so maybe briefly just that I understood correctly. So basically what you have like the slow 5, blow 5 data plus the base call data on the server. And then you would be using like, SAM tools to align the reads to something.
SAM tools to grab the reads from the region, because when there's a remote BAM file, SAM tools already have a feature which can take a URL. And then if you give a region, I want CHR this to this, or even can be a bed file, it will give all the reads that are aligned to the region. So then I take the read IDs and then send through slow 5 curl and get them back.
Amazing. So maybe two brief roll-up ones, and then I give on for other people's questions. But so maybe first, like about the servers where this sort of big data is being stored, right? Like maybe you could explain to us a bit of how you imagine this. You probably know about initiatives like the Squid-based to make the raw nanopore data publicly available. So maybe just having your thoughts on that. And then the second quick question, just at London Calling now, it was announced that nanopore from Q3 on or so, they would switch off the default generation of the raw data, right? Like what your opinion about that is as well. Thank you very much.
So I have a lot of opinions about it. But anyway, like I think like depends on the goal. So if you are doing very large scale sequencing and you have like, you know, bottlenecks with the storage and stuff, of course you can delete it, because if you have money later you can sequence it. So it's a matter of like balancing like the cost of, you know, sequencing versus storage. I think like one thing I have observed is because most people rely on cloud storage now, you ended up paying a huge bill for raw data as well. So that could be the rationale that they say like, okay, sequencing is like cheaper. But in our experience, we have our own NASs in-house, like 1 petabyte of storage we built like 6-7 years ago, like just works. And of course, every like now and then when the hard disk like going bad, the RAID or no problem, we had to swap it. So this has been going great, and like I think it was an initial investment that has been working great. So depending on like how much like people like can afford, and depending on that, I think like that's a choice they can make. But like at least for important samples, I don't think we should go and delete the raw data. For example, we did the like project like which I didn't present, but our team like with the collaboration of many other institutes, this is like indigenous genome sequencing, Australian indigenous population. So those are very like important and hard to find data sets. So they are like archived permanently. And whatever the new base calling modification comes, we might want to go and look at them. So we don't get all those today, right? In the future, we might get them. So for those cases, it's important to store it, I think. But of course, we are routinely, sequencing only for bases, and then not going to look at it. That, yeah, of course, like then it's okay to delete, yeah. So that's what I think. And then for RNA, I think still the accuracy is a bit way to go. So probably necessary to keep too. And also they also announced like protein sequencing. So probably again, like we might have to anyway keep that data because the algorithms will take a while to catch up. Yeah.
So we have a quick question also on YouTube. So the person asks, would Colorado run on AMD NPUs? So these are.
Yeah, so like we like didn't really have access to also NPU is, I think, the neural engine, right? Yeah, no, we didn't implement that. Yeah, it's only implemented for like GPUs at the moment. Yeah, so we have tested on like workstation, like these server GPUs, like MI250 and MI100, and also we have tested on like gaming like card, like I think 7900 XTX or something. I didn't have access to like a laptop GPU to test. Also the Ryzen, whatever, like those mini PCs didn't test. But yeah, NPU we haven't implemented. Yeah.
Hi, thank you for the talk. I was interested in that plot that you showed where you were doing the correlations between let's say the base calling results, even showing that you had some variation between GPUs.
Yeah.
And I noticed that there were, let's say, a few points that were, let's say, in the extreme off diagonals. Yeah. And I was wondering if you have any insights into, let's say, where these.
So we went and did an analysis on that. If you look at the paper, you could like see that analysis. So what we found was it's most of the time the mapper like align it to a different place because it's a very hard like read to map. So because like tools like Minimap, like if you read the documentation, it says that for certain like difficult regions, it like pick something which is not like, like kind of a bit random. So because of that only, that huge identity score variation happened. But when we look at the raw base calls, like the base call versus base call, when we align, they are like 99 or more. So it's like simply that alignment artifact, I believe.
Right, and I guess with nanopore, you also have like this big variability in the lengths of the reads that come in. So if it's a very short read, then also you're going to run into mapping problems.
Yeah, exactly. So yeah, that's true. Yeah. So those kind of outliers were like due to some obvious like reasons like that when we observed. So nothing of concern. Yeah.
Okay, thank you.
With time for one or two quick questions, let's say.
Very nice talk. So I use a lot of your tools, especially Squigulator. So I have a question regarding the Colorado. Is it, so like Dorado, you have multiple base calling modes. Does Colorado also have something like that? So like fast mode and high accuracy?
Yeah, we have support fast mode because these are just models.
Just the models.
Yeah, so we read it like, and then I think we support the V like 4.2 fast hack soup and version 5 fast hack soup. And every model, there is a new parameter or something coming. So a lot of if and else condition if you have to implement. So we couldn't like really go and historically do it. Yeah, because yeah.
Does it also have the server mode of Dorado?
No, we didn't really implement that. Actually, it's not something like, I mean, difficult to implement. Like, yeah, something to consider. Good idea, actually.
Because so I used Dorado with but they only have the Python API for that.
Yeah, which is like a bit like not great. Yeah, I think actually that's a good idea.
Yeah, maybe
Yeah.
might be very helpful for us because
Yeah. I should pass this idea to Bonson. He might not be very happy with you because like you suggested something that he has to do now. Yeah, but thanks for the suggestion. Yeah.
So maybe one last question,
Yeah.
I guess.
So for the lossy part of XZD idea, I was wondering if you tried like some normalization technique that raw signal analysis tools are employing, let's say, instead of straight up like pruning bits, for instance.
Yeah, so like, I mean, yeah, so as I said, like we like tried the worst possible, like, lossy method, like, because we wanted to give the benefit of the doubt to the, to make it worse. So we wanted to show that even with this, like very primitive method, still it seems to be good. So like it's open, yeah. So there should be like wavelet based, all these like, normalized wavelets, lot of fancy methods out there that can definitely do like much better than this. Yeah, that's like, yeah, I think open for exploration. Yeah.
I see. Thanks.
All right. Yes, then let's thank the speaker again. Thanks so much.